Mass spectrometry. In 1900, for the first time J. Thomson introduced mass spectrometer which employed fixed magnetic and electric fields to separate ions of different mass and energy. He recognized that charged particles differing in momentum behaved differently in an electric field, and used this property for separation of ions with different mass. During 1980s its extensive use for research in various fields of biological science was started. During 1990 to 2000, mass spectrometry became an important technique for genomics and proteomics research, leading to the 2002 Nobel Prize in Chemistry to J.B. Fenn and K. Tanaka. Two-dimensional electrophoresis is more powerful when coupled with MS. The unknown protein spot is cut from the gel and cleaved by trypsin digestion into fragments which are then analyzed by mass spectrometer and the mass of fragment is plotted. This mass fingerprint can be used to estimate the probable amino acid composition of each fragment and tentatively identify the protein. The proteome and its changes can be studied very effectively by employing the two techniques together. The MS can also provide valuable information about covalent modification of proteins which can affect their activity. Mass spectrometry is very useful technique. It is used in identification of unknown compounds, quantification of known compounds and determination of structural and chemical properties of compounds when present in small amount 10 to power -6 to 10 to power -8 gram. This technique involves 1. The production of ions of the materials in sample, 2. Their separation on the basis of their mass ratio, charge M-A ratio, and 3. Determination of relative abundance of each ion. Therefore, a mass spectrophotometer consists of three components: the source of ion, analyzer, and a detector. Figure 3.8. It does not directly measure the molecular mass but detects M-A ratio. Mass measured in terms of Dalton Da. 1 Dalton is equal to L12th of mass of a single atom of isotonic carbon-13C+. In recent years, mass spectrometry has become an essential tool for analysis of genome and proteome in its many forms. It is capable of identifying and characterizing proteins present even in picomoles 10 to 12. Basically, it is an analytical device consisting of the following three main components. Figure 3.8. 1. Source of ignition. It involves the gaseous ionization of analyte, the molecule to be analyzed, to be examined. When the molecules gain or lose a charge through electron ejection, protonation or deprotonation, molecular ions are generated. 2. Analyzer. The ions generated as above are separated according to their mass to charge M-Z ratio. 3. Detector. The molecular masses of separated ions are determined from the mass spectra obtained after mass spectrometry of analyte. For identification, protein is separated from the crude extract on a two-dimensional gel. The protein spots are excised and then used as search for mass spectrometry or fragmented into small peptides. Protein fragmentation into peptides is done by proteases, e.g. trypsin. The peptides are separated by using liquid chromatography, such anion exchange affinity or reverse phase column chromatography. Proteins are easily identified if these have been fragmented into peptides. The principle of working of mass spectrometry is to create gas phase ions from polar charged biomolecules like peptides, proteins, or nucleic acids. The sample M undergoes ionization in the ionization source upon introduction in the mass spectrometry. Molecular weight more than 1299 Dalton of the sample results in multiple charged ions, e.g. M+2H. There are many suitable sites of proteins which undergo protonation. All the backbone amide nitrogen atoms and side chain amino group may be protonated. The charged molecules are electrostatically propelled into analyzer. On the basis of M-Z ratio, analyzer separates the ions. The detector detects the ions and transfers the received nulls to a computer. The information is stored and processed by the computer. See figure 3.8. The molecules to be examined should be in the form of gaseous ions. There are several methods available for ionization of biomolecules, but MALDI and electrospray S, each coupled with TOF_MS, are discussed below. 1. Matrix-assisted layer desorption ionization MALDI. MALDI was first developed by Karas and Hillenkamp in 1988. It co-precipitates the large excess of a matrix material with the analyte molecule. In this method, a sub-microliter of the mixture matrix plus analyte pipetted onto a metal substrate and allowed to dry. The dried mixture is irradiated by laser pulse or 7 nanometer, which specifically absorbs the selected solid of matrix. The irradiation causes energy transfer and desorption, resulting in gas-phase matrix ions. With the help of matrix ions, the non-absorbing analyte molecules also get desorbed into the gas phase and ionized. The charged molecular ions of analyte are detected by MALDI TOF mass spectrometer. Generally, MALDI is used for the study of molecules having mass above 500 daltons, i.e. oligonucleotides and peptides. Proteome analysis will be crucial for understanding different biological processes in the post-genomic era. To achieve this objective, mass spectrometry is extensively used for peptide sequencing, identification of protein expression in different tissues and conditions, identification and post-transcriptional modification, e.g. phosphorylation, glycosylation, etc. of proteins in response to different stimuli, and characterization of protein interactions that include protein-ligand, protein-protein and protein-DNA interaction. Different improvement in MS has significantly improved its application in the study of protein structure and function. For the study of macromolecules, the Indian Society for Mass Spectrometry (ISMS) was established in 1978 with its headquarters at BARC, Bhabha Atomic Research Center, Mumbai. This society conducts a workshop each year on mass spectrometry. In India, facilities for MALDI-TOF-MS have been created at several centers, including the Center for Cellular and Molecular Biology, CCMB, Hyderabad, Indian Institute of Sciences, Bangalore, BARC, Rajiv Gandhi Center for Biotechnology, RGCB, Tiruvananthapuram, etc. 2. Surface Enhanced Laser Desorption Ionization, SELDI. It is a unique combination of affinity chromatography and time-of-flight TOF mass spectrometry on a single platform, a combination designated as SELDI TOF-MS, Surface Enhanced Laser Desorption Ionization Time-of-Flight Mass Spectrometry. This system enables protein capture, purification, ionization and analysis of complex biological mixtures directly on protein chips array surface and detection of the purified proteins by laser desorption ionization time-of-flight LDI-TOF. MS analysis. SELDI system consists of 3 parts: 1 protein chip arrays 8 spots of 2 mm diameter, 2 protein chip reader LDI-TOF-MS, and 3 specialized software. 3. Electrospray ionization (ISI). During the late 1980s, Junior Fenn developed ISI spectrometry and was awarded the Nobel Prize in 2002 in chemistry. In this method, the is dissolved in liquid with mobile phase water, acetonitrile, methanol, and pumped through a hypodermic needle at a high voltage, about 4000 volts, which causes strong electric field at the top of nozzle of the metal needle. This disperses electrostatically or electrosprays the highly charged small droplets of about 1 micrometer in size. These droplets are electrostatically attracted to the inlet of MS. Dry gas or heat is applied. The solvent around these droplets soon evaporates and impart charge onto the analyte molecules. The electric field on droplets increases with decrease in their size. Hence, equal charges repel each other. This ionization is very gentle, which causes no breakdown of analyte ions in the gas phase. Fig. 3.9. Protons added during passage through the needle give additional charge to the macromolecule. The MZ of the nucleotide can analyzed in the vacuum chamber. S produces many more ions than MALDI. These ions are transferred into mass spectrometer for analysis with high efficiency. SMS can analyze a wide range of molecules including proteins, oligonucleotides, sugar and lipids. Since a MS measures MZ, it allows high molecular weight proteins to be observed on a low mass range equipment. Thus ISI generates multiple charged molecules. Peptides or proteins are identified on the basis of MZ ratio. The MZ ratio of ions is analyzed after their generation. The new proteins with Human Genome Sequencing Project could be identified through the combination of MS and genomics, and also using the tools of bioinformatics. See Chapter 9. A polymerase chain reaction (PCR). The polymerase chain reaction (PCR) provides a simple and ingenious method for exponentially amplification of specific DNA sequences by in vitro DNA synthesis. This technique was developed by Karya Mullis at Cetus Corporation in Emeryville, California in 1985. Karya Mullis shared the Nobel Prize for Chemistry in 1993. This technique has made it possible to synthesize large quantities of a DNA fragment without its cloning. It is ideally suited where the quantity of biological specimen available is very low, such as a single fragment of hair or a tiny blood stain left at the site of a crime. The details of PCR techniques and its mechanism are described by H. A. Ehrlich in 1989 in his edited book PCR Technology. The PCR technique has now been automated and is carried out by a designed machine. The PCR requires the following. 1. DNA template, any source that contains one or more target DNA molecules to be amplified, can be taken as template. 2. Primers, a pair of oligonucleotides of about 180 to 30 nucleotides with similar G plus C contents act as primers. They direct DNA synthesis towards one another. The primers are designed to anneal on opposite strands of target sequence so that they will be extended towards each other by addition of nucleotides to their 3' ends. 3. Enzyme. The most common enzyme used in PCR is a thermostable enzyme called Taq polymerase. It is isolated from a thermostable bacterium called Thymus aquaticus. It survives at 950 Celsius for 1 to 2 minutes and has a half-life for more than 2 hours at this temperature. The other thermostable polymerases can also be used in PCR. 1. Working mechanism of PCR. The action of PCR involves several cycles. There are 3 steps in one amplification cycles, e.g. denaturation, melting, annealing and polymerization. Extension, figure 3.12. A. Denaturation, melting of target DNA. The target DNA containing sequence between 100 and 5000 bases, to be amplified is heat denatured 950 C for 15 seconds to separate its complementary strands. Step 1. This process is called melting of target DNA. After separation, each strand acts as template for DNA synthesis. B. Primer annealing. The second step is the annealing of 2 oligonucleotide primers to the denatured DNA strands. Since nucleotide sequence of each of oligonucleotide primer is complementary to 3' end of single-stranded template, the primers anneal hybridizes the each template. Primers are added in excess and the temperature lowered to about 680 C for 60 seconds. Consequently, the primers form hydrogen bonds, i.e. anneal to the DNA on both sides of the DNA sequence. Step 2. The annealing temperature varies, but the too low temperature favors mispairing. The annealing temperature in Celsius can be calculated using the formula T is equal to 2 at plus 4 G plus C. C. Primer extension, polymerization, nucleoside triphosphate, DTP, DGTP, DCTP, DTTP, and a thermostable DNA polymerase are added to the reaction mixture. The DNA polymerase accelerates the polymerization process of primers and, therefore, extends the primers at 680 C, resulting in synthesis of copies of target DNA sequence. Step 3. Only those DNA polymerases which are thermostable, i.e. function at the high temperature, are employed in PCR technique. For this purpose, the 2 popular enzymes, Taq polymerase of a thermophilic bacterium, Thymus aquaticus, and the Wnt polymerase from Thermococcus litoralis, are used in PCR technology. These enzymes exhibit relative stability at DNA melting replenishment after each cycle of synthesis. The concentration of magnesium is maintained between 1 and 4 millimole. Thus, in the first step, the target DNA is copied from the primer sites for various distances until the start of second cycle. After completion of step 3 of one cycle, the targeted sequences on both strands are copied and 4 strands are produced. Now, the 3-step cycle, first cycle, is repeated, which yields 8 copies from 4 strands. In the second cycle, the DNA molecules synthesized in the first cycle get doubled. The second cycle is started with heating of double-stranded DNA to result in single-stranded DNA. Each single-stranded DNA again acts as template, i.e. DNA molecules polymerized in first cycle act as template in second cycle. Following the above events, all the single-stranded DNA molecules of second cycle are converted into the double-stranded DNA, figure 3.12. Then third and onward cycles are repeated in the same ways to get more DNA products. The 2 N molecules of DNA are generated using single-stranded DNA as template always after N number of cycles. The third cycle produces 16 strands. This cycle is repeated about 50 times. Theoretically, 20 cycles, each of 3 steps, will produce about 1 million copies of the target DNA sequence, and 30 cycles will produce about 1 billion copies. In each cycle, the newly synthesized DNA strands serve as targets for subsequent DNA synthesis as the 3 steps are repeated up to 50 times. For the working of PCR, about 10 to 100 picomoles of primers are required. The PCR machine can carry out 25 cycles and amplify DNA 105 times in 75 minutes. The number of target molecules should be doubled after each cycle, resulting in exponential increase in the amplified DNA molecules. But the efficiency of exponential amplification is less than 100% due to suboptimal DNA polymerase activity, poor primer annealing, and incomplete denaturation of the template. PCR efficiency is expressed following the formula as given below. PCR product yield is equal input target amount multiplied by 1+% efficiency n, where n is the number of cycles. You can calculate that by using an amplification efficiency value of 70%, about 26 cycles are required to produce 1 gg of PCR product from 1 pg of a target sequence. 10 into 6 amplification, 1 microgram PCR product equals to 1 pg target x 1 plus 0.7 into 26. In recent years, the PCR technology has been improved. RNA can also be efficiently used in PCR technology. The RT-TH DNA polymerase is used instead of the Taq polymerase. The RT-TH polymerase will transcribe RNA to DNA, thereafter amplify the DNA. Therefore, cellular RNA and RNA viruses may be studied when they are present in small quantities. 2. Application of PCR. Some of the areas of application of PCR have briefly been discussed herewith. A. Diagnosis of pathogens. Several pathogens are known that grow slowly. Therefore, their cells are found less in number in the infected cells' tissues. It is difficult to culture them on artificial medium. Hence, for their diagnosis, PCR-based assays have been developed. These detect the presence of certain specific sequences of the pathogens present in the infected cells' tissues. Besides, it is useful in detection of viral infection before they cause symptoms or serious diseases. B. Diagnosis of specific mutation. Presence of a faulty DNA sequence can be detected before establishment of disease. By using PCR, sickle cell anemia, phenylketonuria, and muscular dystrophy can also be detected. The other diseases can also be diagnosed by using PCR. For example, PCR_based diagnostic tests for AIDS, chlamydia, tuberculosis, hepatitis, human papillomavirus, and other infectious agents and diseases are being developed. C. Prenatal diagnosis. It is useful in prenatal diagnosis of several genetic diseases. If the genetic diseases are not curable, it is recommended to go for abortion. D. DNA fingerprinting. DNA fingerprinting is most successfully used in forensic science to search out criminals, rapists, solving disputed parentage, and uniting the lost children to their parents or relatives by confirming their identity. This is done through making link between the DNA recovered from samples of blood, semen, hairs, etc. at the spot of crime and the DNA of suspected individuals, or between child and his/her parents' relatives. A. In research, DNA fingerprinting of new microorganisms isolated from various extreme environment, soil, water, sediments, air, extreme habitats, etc. is also carried out to confirm their identity by comparing with the DNA sequences of known microorganisms. Their DNA and RNA can be amplified. It is also useful to determine the orientation and location or restriction fragments relative to one another. F. In molecular archaeology, paleontology, PCR has been used to clone the DNA fragments from the mummified remains of humans and extinct animals such as the woolly mammoth and dinosaurs from the remains of ancient animals, as recently epitomized in Michel Crichton's Jurassic Park. DNA from buried humans has been amplified and used to trace the human migration that occurred in ancient time. G. Diagnosis of plant pathogens. A large number of plant pathogens in various hosts or environmental samples are detected by using PCR, for example, viroids associated with hops, apple, pear, grape, citrus, etc., viruses such as TMV, cauliflower mosaic virus, bean yellow mosaic virus, plum pox virus, putiviruses, mycoplasmas bacteria, Agrobacterium tumefaciens, Pseudomonas solanacearum, Rhizobium leguminosarum, Xanthomonas campestris, etc., fungi, e.g., Collectotrichum gliosporioides, Glomus spp, Laccaria spp, Phytophthora spp, Verticillium spp, and nematodes, e.g. Meloidogaon incognita, M. javanica, etc. There are some other PCR-based techniques. 3. Reverse transcription-mediated PCR, RT-PCR. Amplification of double-stranded DNA can also be applied to produce double-stranded cDNA also, which is synthesized from mRNA using the enzyme reverse transcriptase. Based on this principle, RT-PCR was developed. RT-PCR includes a single application combining the process of cDNA synthesis by reverse transcription and PCR amplification. Using this principle, one can isolate very rare mRNA transcripts from cell samples through RT-PCR. The thermostable enzyme RTTH uses RNA templates from cDNA synthesis and thus allows single enzyme RT-PCR viral reverse transcriptase RTase from avian murine virus AMV RTase or mammalian leukemia virus MLV RTase can also be used for cDNA synthesis. Both the enzymes IE RTase and RTTH DNA polymerase are used alternatively in the RT-PCR. RAPD markers have been successfully used for 1. Construction of genetic maps e.g. Arabidopsis pine, Helianthus, etc. 2. Mapping of traits. 3. Analysis of genetic structure of population. 4. Fingerprinting of individuals. 5. Identification of somatic hybrids, etc. A detailed account of RAPD is given in Chapter 14. 5. Arbitrary primed PCR, AP-PCR. In 1990, J. Welsh and M. McClellan developed the AP-PCR and carried out fingerprinting of genomes with arbitrary primers. It is a type of RAPD where single primers of 10 to 50 bases are used to amplify genomic DNA in PCR. In the first 2 cycles, annealing in under non-stringent conditions. AP-PCR differs from RAPD in some respects. 1. In AP-PCR, amplification occurs in 3 parts, with its own stringency and concentration of constituents. 2. Higher concentrations of primers are used in the first cycles of AP-PCR. Primers of different length are arbitrarily selected, e.g. Me3 universal sequence primer. 3. Products of AP-PCR are analyzed on acrylamide gels and detected by autoradiography, whereas these of RAPD are analyzed on agarose gels, stained with ethidium bromide and visualized under UV light. Problem statement. End of the Chapter 3.